Have you ever run into one of those problems where you just can't get two things to work together properly? Maybe it's a medicine that won't dissolve, or it tastes just awful, or maybe it goes bad way too quickly. These aren't just small annoyances, right? They can be real roadblocks in getting effective treatments or products out there. So today we're doing a deep dive into a really fascinating molecular solution for exactly these kinds of challenges. We're talking about a unique molecule called cyclodextrin and the art and science of making what we call cyclodextrin complexes.
That's right. And we've gathered quite a bit of information. Some of it gets, pretty technical. But our goal today is simple. Give you a clear, accessible picture. We want to break down how these amazing complexes are actually made, why they're so useful, and importantly, how scientists make sure they actually work as intended. You should walk away with a good handle on this specialized but really impactful area of science.
Okay, let's get into it then. So picture this, like a tiny molecular donut. That's kind of our cyclodextrin, right? What's so special about this little donut shape?
Well, that shape, that truncated cone or donut, it's absolutely key. The outside part likes water. It's hydrophilic. But the inside, the cavity, that's hydrophobic. It repels water. And because of that, it really loves to invite other hydrophobic molecules, especially ones that don't mix well with water, to sort of tuck themselves inside.
Ah, okay. So it's like a tiny hideout, a safe space for molecules that don't like being surrounded by water. They can just slip inside this dry pocket.
Exactly. A molecular hideout, that's a good way to put it. And this complex formation, it isn't static like a key in a lock. It's actually a dynamic thing, an equilibrium. The active ingredient, the API, the bit that does the work, is constantly moving in and out of that cyclodextrin cavity. So when we design these, our main job is to push that balance, that equilibrium, towards more complex being formed. That's how we get the benefits.
Right. So given this hideout ability, what are the immediate wins? When a molecule gets into that cyclodextrin donut, what problems does it solve for us, practically speaking?
The advantages are pretty profound, actually. They hit some of the biggest hurdles in product development. Think about, say, bad taste or smell, skin irritation, even toxicity. Cyclodextrins can mask those quite effectively. But it goes way beyond masking. They can dramatically improve key physical properties, for instance, solubility. The complex generally takes on the cyclodextrin solubility, which can be a massive boost for an API that barely dissolves in water. This is huge for making certain drugs usable. And they also improve stability, protecting sensitive stuff from breaking down. They enhance wettability, which is how easily a powder mixes into liquid. And critically, they can significantly boost bioavailability.
Bioavailability. That's how much of the drug actually gets into your system and can work, right?
Precisely. And it's worth noting, cyclodextrins aren't just one single molecule. They come in different sizes, and importantly, with various chemical modifications. Modifying those oxygen groups can seriously improve their solubility and how they function, often making them much better than the basic native ones.
Sounds like a really powerful toolkit. How widely are these actually used? Are we talking niche applications or?
Oh, it's quite established. Right now, there are 9 different cyclodextrins approved for use in pharmaceutical products. 7 of those are used with small molecule drugs, usually as excipients, the inactive ingredients that help deliver the drug. Then 1 is used with biologics, as a vaccine adjuvant, something that boosts the immune response. And interestingly, 1 cyclodextrin is even approved as an active drug itself. And beyond pharma, they're everywhere. Case masking in foods, ensuring content uniformity for really potent drugs where dosage has to be exact, improving dissolution for things like painkillers that need to act fast, chemical stabilization, as we said. What's really neat is how flexible they are. They work in solids, liquids, semi-solid sprays, even chewing gums. They're incredibly formulation friendly.
Okay, theory's great, but let's talk specifics. Can you give us some real-world examples, maybe some products where cyclodextrins made a huge difference?
Sure. There's hofoxolone, a veterinary drug.
Yeah.
Cyclodextrins were key to getting it soluble enough in water to be effective. Or think about the glucagon nasal spray from Eli Lilly. That showed something like a 7 to 8-fold jump in bioavailability thanks to cyclodextrins. That's huge.
Wow, 7 to 8 times.
Of turning an injectable into a nasal spray.
Yeah.
And shelf life. We've seen products go from lasting weeks to being stable for years. And a really crucial point is that these benefits often stack up. Improving solubility might also make the API more stable and boost its bioavailability all at the same time, especially if it degrades quickly.
Okay, but there's got to be a catch, right? You mentioned these benefits only come if you get true complexation. What if you just mix them?
Yes, the true complex problem. That's a real issue. Especially you see it sometimes in nutraceuticals.
Right.
Products might list cyclodextrin, but if you test them, no actual complex is formed properly. And if that happens, you miss out on all those potential benefits. You're essentially just adding an expensive filler material that doesn't do the job.
And the remdesivir example you mentioned really drives this home.
Absolutely. Remdesivir, for COVID-19, uses cyclodextrin for solubility. Now, if you just physically mix the cyclodextrin powder and the remdesivir powder same amounts as in the approved drug, it doesn't work well. The cyclodextrin dissolves, sure, but the API stays largely insoluble because it hasn't properly entered the cavity. But take that exact same mixture and process it correctly, like freeze-drying it, lyophilization, or using electrospinning, then suddenly it's perfectly water-soluble. Why? Because you force the formation of a true complex. That difference in processing is everything. It's a big aha moment.
Okay, so we definitely see why making a true complex is so important. The benefits are clear. But let's get into the nitty-gritty. How do you actually do it? This sounds like where the real deep dive begins.
Right. So designing and making a cyclodextrin product, it's usually a pretty systematic process. First step, you have to define your goal and limitations. That means looking hard at the API itself, its structure, its properties. Then, how will it be administered? Oral, .4 topical. And what's the final form? Tablet, liquid. All this helps narrow down which of those 7 main pharma cyclodextrins might work. Regulatory factors also come into play early on.
Makes sense. What's next?
Second, you select the manufacturing technology. This choice really matters because it determines which solvents you can use, if any, and that influences which cyclodextrins are suitable. Third, and this is critical, experimentation. You have to screen different cyclodextrins in the lab. Yes, there are prediction tools, even AI models that can suggest how strongly things might bind, but they're not foolproof. Actual lab trials are still the gold standard to confirm what works best for your specific ATI and goal. You can't just guess.
So hands-on testing is unavoidable.
Pretty much. And finally, you need to actively influence that equilibrium we talked about, push it towards complex formation. There are several levers you can pull. Things like cyclodextrin concentration, the pH of the solution, the temperature, how long you stir it, and ideally, you want long stirring times, maybe 8 to 24 hours, to really reach equilibrium. Stirring speed matters too.
Yeah.
And of course, the molar ratio, how many cyclodextrin molecules per API molecule. You tweak these to get the best result.
That's quite a few variables to manage. Now, beyond just the cyclodextrin and the API, is there anything else you can add to the mix to sort of supercharge the performance?
Oh, absolutely. That's where additives or co-components come in, and they are really important. Most commercial cyclodextrin products aren't just 2 ingredients. They often contain other things, maybe hydroxy acids, amino acids, certain types of polymers that create a synergistic effect. They work together with the cyclodextrin to boost performance way beyond what either could do alone.
Synergy. So the sum is greater than the parts.
Exactly. And what's fascinating and sometimes challenging is that this synergy is often unpredictable. You can't easily calculate it. It really requires careful experimentation to find the right combination that gives you that extra boost.
Do you have an example of that, where adding something extra made a huge difference?
Yes, the domperidone case is a classic. So 10 milligrams of domperidone normally need something like 2 liters of water to dissolve. Just imagine that.
2 liters.
Wow. Right now, add cyclodextrin alone, and you get that down to about 62 milliliters. Better, but still a fair bit. Add a specific hydroxy acid alone, and it's about 12 milliliters. But, and here's the synergy, combine that same cyclodextrin and that same hydroxy acid, and suddenly you only need 0.3 milliliters of water to dissolve the 10 milligrams.
0.3, that's incredible. From 2 liters down to less than half a milliliter.
It's a massive difference. It shows how powerful finding the right co-components can be. And there's another layer too. The form of the API itself matters a lot. Are you using the free-base form? Or a particular salt form? This choice affects the drug's basic intrinsic solubility, obviously, but it can also dramatically affect how much the cyclodextrin can improve that solubility. You might have 2 forms of an API where one is intrinsically twice as soluble as the other, but once you complex them with cyclodextrin, that difference might blow up to a 6 or 7-fold difference in final solubility. So picking the right salt or base form right at the start is also crucial for getting the best out of the cyclodextrin. Definitely. It all feeds in. Choosing the right cyclodextrin is vital, yes, but optimizing the whole formulation around it, the co-components, the API form, that's often where the real magic happens. Okay, so we figured out how to form the complex, how to make it better with additives. Now, how do we actually get it out of the solution or mixture so we can put it into a pill or a liquid? We need to isolate it. There are quite a few ways, depending on what you start with. If it's in a liquid, a common approach is the solution method. You dissolve both the cyclodextrin and the API together, let the complex form, and then isolate it. Often that's done by lyophilization, which is freeze-drying. It's gentle, good for sensitive things, but can be expensive. Another liquid technique, maybe for APIs that don't like water, is co-precipitation or co-evaporation. You might mix an organic solution of the API with a water solution, the cyclodextrin, form the complex at the interface, and then remove the solvents.
Okay, what about if you're not starting with everything dissolved, like solids?
Right, you can also work from solids or semi-solids. On a lab scale, you might use literally mixing it like dough with a small amount of liquid. Industrially, that scales up to extrusion. These methods use physical force, shear, to help form the complex. They're efficient for large amounts and avoid lots of solvent, but maybe don't achieve quite as complete complexation as solution methods sometimes. You need good process control. And then there's also grinding or milling, again using physical force, but more in a powder state. Each technique has pros and cons depending on the drug and what you need the final product to be.
So lots of options for making and isolating, but then comes a big question. Once you have this powder or solid, how do you prove you actually made the complex? How do you know it isn't just, the original stuff mixed together? This sounds like where the lab coats really come out.
Exactly. You need analytical proof. Basically, any method that can tell the difference between the free API and the API tucked inside the cyclodextrin will work. A couple of key ones are DSC, differential scanning calorimetry. Think of it like taking a thermal fingerprint. A pure crystalline drug has a sharp melting point. When it forms a true complex, that sharp peak usually disappears or shifts dramatically because it's not really a crystal anymore. It's now part of this amorphous complex.
So the melting behavior changes. What else?
X-ray diffraction is another big one. Crystalline materials give sharp diffraction patterns. Amorphous materials, like most cyclodextrin complexes, give broad halos. So if the API's crystal pattern vanishes, that's strong evidence of complexation. Then you have techniques like HPLC, high-performance liquid chromatography. It separates components. The complex API will usually travel through the HPLC column differently than the free API, so you see a shift in its elution time. And for really direct proof, NMR, nuclear magnetic resonance spectroscopy, can be powerful. It can actually show interactions between the API protons and the cyclodextrin protons, giving evidence of inclusion and even hints about the geometry, how it's sitting in there. Though the cyclodextrin signals can be strong and sometimes complicate things.
So NMR gives a closer look.
It can, yeah. And other methods like ITC, isothermal titration calorimetry, which measures heat changes during binding, and UV spectroscopy are also very useful tools in the arsenal. The point is, you need solid analytical data to confirm you've actually made what you think you've made.
Got it. So it's not just if it formed, but confirming it's the real deal. What else do you need to figure out about the complex once you know it exists?
Well, several other things are important to assess. Stability is a huge one. Physical stability, thermal stability, chemical stability. Does the complex protect the API while it's sitting on the shelf? Solid complexes can be very stable, often for years. But it's important to remember, in vivo, once it's in the body, the complex usually needs to dissociate to release the API so it can work. So while the complex protects on the shelf, protecting the released API from things like enzymes in the body is a different challenge.
Right. Shelf stability versus stability in the body. What else?
You often want to understand the binding strength. How tightly is the API held? Interestingly, very strong binding isn't always necessary or even desirable for a successful product. Sometimes weaker, transient binding is fine, but knowing the strength helps optimize things. Then there's stoichiometry. What's the molar ratio? Is it 1 cyclodextrin per API molecule? 1.1, or maybe 1.2, or 2.1. This is key for formulation. And crucially, you almost always need an excess of free cyclodextrin in the final system.
An excess? Why is that?
Because of that equilibrium we keep mentioning. The API naturally wants to pop out of the cavity sometimes. Having extra free cyclodextrin around helps push the equilibrium back towards the complex state, keeping the API solubilized or stabilized. So the actual molar ratio in a formulation might be 1.1.4 API to cyclodextrin, or even up to 1.15, 0.7 in some cases, to ensure enough free cyclodextrin is present.
Okay, so you need more donuts than guests, essentially.
Kinda, yeah. Another aspect often asked for by regulatory agencies is the geometrical assessment. How does the API actually fit inside the cavity? Which part goes in? How deep? This helps prove it's a real inclusion complex and understand its properties. Computational modeling is really useful here. And finally, dissociation kinetics. How quickly is the API released from the complex? This affects how fast it can cross membranes or bind to proteins in the body, influencing its overall action.
This is all incredibly detailed. It leads me to wonder, thinking about generic drugs. Can a company just decide to add cyclodextrin to make a better generic version of a drug that didn't originally have it?
That's very, very difficult. I'd say almost impossible from a regulatory standpoint for most standard generic pathways. Why? Because adding cyclodextrin fundamentally changes the drug product. It alters solubility, possibly the onset of action, the whole pharmacokinetic profile, how the drug moves through the body. So proving that your new cyclodextrin-containing version is bioequivalent to the original non-cyclodextrin version becomes extremely challenging, if not impossible.
So it's not really a viable generic strategy.
Generally, no. There are very few, if any, examples of that succeeding in the market. You're essentially creating a new drug product which requires a much more extensive development and regulatory process. Also, there's the issue with the FDA's Inactive Ingredient Database, IID. Only certain cyclodextrins are listed there with established safety limits for different routes. If you want to use a cyclodextrin that's not listed, or use a listed one by a route it's not approved for, especially something like IV, you're likely facing many more regulatory questions and needing extra safety studies. It adds significant hurdles.
Right, that makes sense. It's not just a simple swap.
Not at all. So maybe let's try and boil down the key messages from this whole discussion. What are the absolute must-dos? First, always pick the best cyclodextrin through actual lab trials. Don't just rely on theory or what worked for something else. Test it for your API. Second, fine-tune the formulation with co-components. Don't just assume a simple binary mix is best. Experiment to find those synergistic effects that can really optimize performance. Third, choose the right isolation technique for your specific API and product goals. How you make it matters. And maybe most importantly, confirm that complexation actually happened using proper analytical tools. Make absolutely sure you have a true complex in your final product, not just a simple physical mixture. That's the foundation for getting all the benefits.
Fantastic summary. It's pretty amazing to think how this tiny molecular donut, this cyclodextrin, can completely change the game for medicines and other products, how they work, how they're delivered, how long they last. It really highlights the power of designing things at the molecular level. It makes you wonder what other sort of hidden molecular superheroes might be out there just waiting for us to figure out how to use their full potential. What common problems do you, listening now, run into that maybe, just maybe, a clever bit of molecular engineering could solve one day? Something to think about. Thanks for diving deep with us today. Until next time, keep learning.